This talk will describe a software tool for visualization of organism-scale metabolic networks. The motivations for this work are that multiple life science areas require information on organism-scale metabolic networks, including metabolomics, microbiome research, synthetic biology, and basic research. Overall, genome sequencing plus computational metabolic reconstruction methods have yielded metabolic networks for thousands of organisms. And to fully understand and exploit these reconstructions, visualization of whole metabolic networks is critical. I'll define the metabolic network visualization problem as how to enable visual analysis of full metabolic networks. Because these networks are so large, semantic zooming is almost certainly required to solve the problem. And also because of their large size, searching algorithms the ability to search the diagrams are important. And we want these diagrams to avoid large complex hairballs that are visually very difficult to understand. We want to be able to visualize metabolic networks not only from single organisms, but from pangenomes and from organism communities. We want to be able to overlay high throughput data onto these diagrams, such as transcriptomics data, and we want to be able to use these diagrams to compare networks from multiple organisms. Now I'm going to survey three different overall approaches that have been taken to this problem. One is to print metabolic wall charts as posters. These metabolic posters have aided and inspired scientists for decades. For example, Roche and Sigma Aldrich both make these posters available. Some of the limitations of printed charts are that they're time-consuming to update, they're difficult or impossible to search, and it's unclear how they integrate information from many organisms. For example, it's often very difficult to tell in these diagrams which pathways are present in exactly what organisms. They also cannot be used for analysis of high throughput data or for comparative analysis or for microbiomes. The second approach is what I will call the Uber chart approach, and this approach is used by KEGG. The idea is to create one or a small number of large diagrams that contain a fixed set of pathways, and thus do not require automated layout algorithms. Because there's such a small number of diagrams, the diagrams can be arranged by hand, although that will still be time-consuming. So it is good to have layout algorithms. So for example, KEGG provides 6 different Uber charts for different areas of metabolism. Here I'm showing their Uber chart for secondary metabolism. One of the main limitations of this approach is that the majority of space is consumed by pathways that are not in a given organism of interest. For example, E. coli, which has 364 metabolic pathways in EcoPsych, that's only 12% of the 3,000 pathways in our MetaPsych reference pathway database. And so since the Uber charts typically contain all known pathways in a given database, this means that the vast majority of the diagram that we're looking at is not actually relevant to E. coli. Now the final approach is the approach taken by BioCyc, and it is to create organism-specific metabolic chart diagrams, where each chart contains pathways from a single specific organism. And thus, because one could want to analyze thousands of organisms, one needs automated layout algorithms that generate charts for each organism. That's the approach we take in Pathway Tools. And here we're looking at the pathway diagrams for E. coli and for humans, which, as you can see, have fairly different content. Now, before I get deeper into metabolic network diagrams generated by Pathway Tools, let me give you the big picture in terms of what Pathway Tools is and how it operates. Pathway Tools is a large bioinformatics software system that can take as input an annotated genome. It can run its pathway inference component called pathologic and create a pathway genome database containing the genome plus the inferred metabolic network of the organism. That database can then be input to several different tools, including Metaflux for creation of metabolic flux models, pathway genome editors for updating and modification of these databases, and the Pathway Genome Navigator, which contains search and visualization tools, including the metabolic network tool that I'll describe. Pathway Tools has many other capabilities. It has genome informatics capabilities, as well as pathway informatics, regulatory informatics. It has a number of microbiome operations. It performs comparative analysis, transcriptomics and metabolomics data analysis, and various advanced data manipulation. Now, we've used Pathway Tools to create the BioCyc collection of more than 20,000 pathway genome databases. And each of these databases has the same schema and contains pathway data types and proteome and genome data types. And we divide these 20,000 databases into three groups. The Tier 1 and Tier 2 databases are highly curated and moderately curated, respectively. And the Tier 3 databases, all the rest, are computationally derived. Both BioCyc and KEGG contain a set of reference pathways that they use to predict the pathways present in a given organism. And for BioCyc, that reference database is called MetaCyc. MetaCyc is one of the databases in the BioCyc collection. MetaCyc has 3,000 metabolic pathways compared to the 400 metabolic modules present in KEGG. MetaCyc has 17,000 reactions compared to the 11,000 in KEGG. They both have about 18,000 metabolites. And both databases provide curation of what are called mini-reviews, namely curator-authored summaries of enzymes and pathways. MetaCyc has about 11,000 textbook page equivalents of these mini-reviews. KEGG has about 1,500 textbook page equivalents. So you can see MetaCyc has much more in-depth curation than KEGG does. Let me now take you through the steps in creating Pathway Tools metabolic network diagrams. Now, the input to this visualization algorithm is a pathway genome database for the organism that contains all of its metabolic pathways and reactions. And the first thing that the metabolic network diagram software does is to query this pathway genome database to determine the cellular architecture of the organism. Namely, what are the major spaces, compartments present in this cell type, such as the periplasm and the cytoplasm. The next thing the software does is determine the set of reactions, proteins, and pathways present in these major cellular spaces. It then lays out each individual pathway diagram using the Pathway Tools layout algorithms. It then runs a layout algorithm that arranges the pathways into what we call pathway blocks. And in this example diagram, here's an example pathway block, namely all the pathways in E. coli involved in amino acid biosynthesis. Another pathway block would be all the pathways in E. coli involved in carbohydrate degradation. The next thing the software does is to arrange the pathway blocks relative to one another within three major regions of the diagram that capture biosynthesis, catabolism, and energy metabolism. So for example, the amino acid biosynthesis block is part of this larger biosynthetic pathway region. And finally, the software lays out grids of individual reactions and arranges transporters in the cellular membrane. Here's the grid of individual reactions. There's one for the cytoplasm and there's one for the periplasm. And here are the transporters in the cell membrane. Now, the layout and image generation for the metabolic network diagram occur in the Common Lisp Pathway Tools server. And the server creates the image as a JSON structure containing all the graphics, all the primitive graphics operations that together make up the chart. And in fact, the software generates 4 different versions of the chart graphics at different semantic zoom levels. And then those 4 versions are sent to a JavaScript engine in the user's web browser for display. And the user zooms between different levels and can perform searches in the web browser. And we can also translate the graphics to PDF to create printable metabolic posters from these diagrams. And to show you what the zooming looks like, here's the E. coli metabolic chart, and each dot is a metabolite, each line is a metabolic reaction, and as I move my mouse over them, they're identified. Then this ladder can be used for zooming, and I can use my mouse wheel for zooming. So I'm zooming now into the amino acid biosynthetic block, and as the diagram gets larger, we begin to see new elements, namely the metabolites in each pathway, the names of each pathway, and eventually we will see the enzyme and gene names at each metabolic reaction. And as you can see, the zooming happens in real time. It's quite fast. We can move the diagram around, zoom in on other regions, and perform search. The search commands cover searches for genes, metabolites, etc. And in this particular diagram on the right, what we've done is to search for all E. coli genes regulated by the FNR transcriptional regulator, which is an oxygen-related regulator. We can also use similar commands to perform species comparisons to highlight reactions that are shared or not shared between specified organisms. Now, if we have pathway genome databases for every organism in a microbial community, we can create this kind of diagram which shows the cellular overviews for every organism in the community. Here we're showing the altered Shadler flora, which is a mouse microbiome, and we've highlighted in different colors the pathways that are unique to each organism in the community. To use the diagram for omics data analysis, we can import, say, a transcriptomics data set in a tab-delimited file, map the transcriptomics values to a color scale, and then color each reaction in the diagram with a color representing its expression level. And we can zoom the diagram the same way you just saw with the highlighting, and we can actually play the diagram, an animated version of the diagram, to show how the diagram looks at different time points. And the user can click on reactions in the diagram to bring up an associated graph of the transcriptomics values at each of the 6 time points for that reaction. So in summary, Pathway Tools has generated thousands of these organism scale metabolic map diagrams. The diagrams are organism specific, they're searchable, they're zoomable, they can be overlaid with omics data, they can be used for comparative analyses, and they can be used to show metabolic interactions within organism communities.